Processes of Recombinant DNA Technology, Part 2. Formation of chimeric DNA. Vector DNA is cleaved at specific sites with the same restriction endonuclease which is used to cleave donor DNA to form sticky ends. Complementary sticky ends of donor DNA and vector DNA are joined by the enzyme ligase to form hybrid DNA or rDNA or chimeric DNA. Uptake of chimeric DNA. Chimeric DNA is introduced directly by using calcium chloride or by the process called phage-mediated transduction. Selection of cells containing chimeric DNA. The transformed bacteria are identified by using genetic markers or by colony hybridization technique. Genetically engineered bacteria are propagated in large quantities and the product formed by the donor gene is recovered. Amplification of gene of interest using PCR. Polymerase chain reaction helps in multiplying the genes in more number. 3 major steps in PCR are repeated for 30 to 40 cycles. The instrument used for conducting PCR is called automated thermocycler. The reaction mixture contains primers, DNA to be cloned, DNA polymerase enzyme, and DNTPs. Stepwise procedure of PCR. Denaturation at 94 degrees Celsius. Double-stranded DNA is subjected to denaturation by breaking up of hydrogen bonds. Short chains of single-stranded DNA are formed. Annealing at 54 degrees Celsius. As primers move around in a Brownian motion, bonds are established between single-stranded primer and single-stranded template. The primers, which are stable after attachment to the template, initiate the copying of template with the help of polymerase. Extension at 72 degrees Celsius. Polymerase requires an optimum temperature of 72 degrees centigrade. Bases form couples with primer on 3' sites and DNTPs are added from 5' to 3'. Some primers which have no exact match do not synthesize any extensions.